What's that? I like this weather if you're staying at home. Hot chocolate. What's that? You're dead? Why are you dead? Dead?
I have a question for the vascular.
Question for the vascular, way back when?
You
What's up?
said that, so I wrote originally that veins have a lot more smooth muscle than
No,
arteries.
no, veins do not have a lot.
That's exactly what you said, but then I went to this slide and it doesn't say that.
I would never say that veins have more
Okay,
smooth
so
muscle. Never,
clarify.
ever, ever.
Arteries have
Says
more.
arteries have more smooth muscle. Yeah, that's all about controlling blood flow with circulation.
Okay.
Actually, I have one section for ADH when it has to go with water. When it is filled with water,
Okay, so you missed it. I just asked them about
sodium doesn't do anything.
No.
veins.
Just the
He
water.
said arteries. What? Arteries.
That'll make more sense when we get to kidneys.
That have a smooth muscle? Yeah.
Ultimately, remember, water is a polar molecule, right? So how does it get to a membrane? It needs a channel. ADH binds to a G protein receptor that makes water channels. They even have a cute little name. The water channels are aquaporin. Make sense? All right. Any other questions?
We haven't started. Oh, we're not having started.
Not okay. I think we've pretty much covered this. Did I miss something on this one? I think we ended up with, again, over here on the active side, you create an immune response either from some sort of exposure, naturally occurring, i.e. you get a cold, or through vaccines. We'll talk more about that in a little bit. Okay, so ultimately when you're an adult, you have hopefully a nice developed acquired immune system, and that's going to work with your innate immune system or defense. So again, your body's defenses, we have the basic external things, skin, hair, mucus, secretions, you name it. We're not too concerned about that. So we got a little of that in the integumentary. We are going to be concerned with what happens if they break through your physical barriers, and now we are in an internal war, if you will. And that's going to involve several different mechanisms as we put this on. So your innate, like I said, outside external defenses, we have phagocytosis. You can eat bacterium, dead cells, things of that nature. We have the inflammatory response, natural killer cells. Is that the coolest name ever for a cell? Natural killer cell. The complement system, which are antimicrobial proteins, and interferon, which is kind of an alert that, hey, bad things are out there. So we're going to dissect each one of these. So phagocytosis, again, we can destroy abnormal cells, bacterium, things of that nature. Three key things. We have margination. We can have these phagocytes, if you will, just hang out there inside the lining of the capillaries waiting to see something that doesn't belong. Or it can leave the capillaries and go into the interstitial fluid through a process called diapenesis. Diapenesis. They will leave the walls or the inside of a capillary and exit through the squamous cells, between the squamous cells, if they're drawn to a site, and that is called chemotaxis. Chemotaxis. Chemo for chemical, taxis, think of taxi. Right? So infected cells, damaged cells will release what are called chemotaxins, and these are chemical signals, and that will draw other phagocytes to the area for the big battle. Dun, dun, dun. So again, we have our white blood cells that have the phagocytic ability, Dendritic cell, you may remember that, probably not, from the integumentary system. Monocytes, macrophages. So, quick overview, we have monocytes. They can differentiate or develop into macrophages, large phagocytic cells. It takes a while to get to the site of the infection compared to neutrophils, but when they do, they are constantly multiplying, mitosis, until you have a whole host, an army. These monocytes and macrophages hang out on their lymph nodes, spleen and lungs, looking for, again, infected cells, cancer cells, bacterial infections. We have the macrophages, which are their monocytes, and then we have microphages, which are much smaller. So we have neutrophils. Again, we covered this in blood. They get their names just because of how they die and they stain. Functionally, neutrophils are plenty. What percent of your white blood cells are neutrophils normally? 70%. So a lot of them there, and they have a whole host of weapons. They can use lysozymes, releasing enzymes that break down bacterium. They have proteins that act like antibiotics. Who's taken microbiology? How does an antibiotic work? How does good old-fashioned penicillin or erythromycin or amoxicillin, how do they work? What's their key action?
Cell walls.
Cell walls. Very good. Outstanding. Attack the cell wall. They also have oxidants like bleach-like chemicals that can destroy cells. Eosinophils are key against parasitic worms, larvae, small. They also can phagocytize our antibody-antigen complex. Remember, antibodies bind cells together like we saw in our agglutination lab. And once we bind our pathogen, the eosinophils can come in there and eat the whole darn thing. And again, the dendritic cells found mostly in the integument, in the epidermis, also in the phagocytes. Again, there's nothing significant about this. It's just an overview. They engulf our microbes, a lysosome, a vesicle with enzymes in it, merges, and they just start to break it down. And this is the key slide right here. This is a highly testable. You gotta make sure that what we're eating isn't the good guy. So again, three things. We have chemotaxis. Again, damaged, infected cells. We release these to draw our phagocytes to the area. And then we have something called opsonization. Opsonization. And that is putting markers on the target cell to identify it as a bad guy. Proteins are called opsonins. So you just need to remember opsonization is marked for death. Does that ring a bell to anybody? Nothing? No one just heard marked for death? Good old Steven Seagal movie from the 70s? Nothing, huh? All right. So again, we attach these proteins to the cell that identifies it as a bad guy. Then on the phagocyte, we have receptors that will bind to those opsonins, and that will be the key to trigger the phagocytic activity. Knowing that this is definitely a bad guy, and I'm going to destroy it. So our TLRs, toll-like receptors, are receptors on the phagocyte. You're going to bind to receptors on the target cell, and that will trigger the engulfing process. Then we have the inflammatory response. We kind of think of inflammation as a bad thing, don't we? We're always trying to stop inflammation. What was the point of inflammation? Evolutionary. You got any ideas?
Tell you something wrong.
What's that? Okay?
Mm.
You don't know? Okay, again, you have all these white blood cells in your circulatory system, and if you have an infection or damage, we need these cells to be able to get out of the circulatory system and into the tissues and the interstitial fluid. So the inflammatory response was make the capillaries really porous, really leaky, so these cells can get out. Of course, the fluid leaves as well, causing the swelling. Barrier, not the greatest mechanism that we've evolved with, because it assumes that there's damage. That damage is open to the environment, and there may be an infection, and we need the white blood cells. But if I sprain my ankle, what's the point of inflammation? Nothing. Nothing but pain in the butt. So we do what? Rice. Rice. Yeah, we do it in our power to get rid of the inflammation. So today's world, most of inflammation is a bad thing. In fact, you hear somebody that falls off the horse and is paralyzed, you know, paralyze them 90% of the time. You get a spinal injury, that area gets inflamed, and it's the inflammation that finishes off paralysis part, not the actual. So nowadays, especially at football games, what do they have right there on the sideline in case they have a neck injury? No one knows? Huh? Paramedics. Yeah, paramedics guys. They have an ice bath. If they suspect somebody that's got the injury, they can immediately ice them to keep that inflammation down.
Unless you go to University of Maryland.
What?
University of Maryland, that whole thing, whole place,
Yeah, I didn't do that.
yeah, football coach.
Did it? Yeah, see, that happens. So again, the inflammatory response starts with our macrophage and mast cells releasing histamine. Right? And histamine is a chemical signal that causes vasodilation, and it opens up those capillaries, enlarging the gaps, so again, cells and fluid can get out. Everything else, the swelling, the redness, all that is just incidental to this whole process. And that's all you need to know. Next mechanism is our natural killer cells. And what they do is bind to an infected cell, cancer cell, pathogen, and destroy it. And it produces proteins that are called perforins. Perforins. Guess what a perforin does? Very good. Perforates. Puts holes in the target cell. And I like the artist that, what is this? There's their Golgi complex saying we're going to make these proteins, these perforins, and bringing them to surface. And basically, a perforin just puts a hole in it. What makes the cell go boom? What causes the cell to explode? Come on, Candace. If they put a hole in a cell, why would it blow up like that? Water. Osmosis. See, even the cells know about osmosis. So yeah, just put holes in it and let water do its thing. The next thing you have is our antimicrobial proteins, the complement system. Quite a few, find more all the time. And they involved in a couple different things, as we're going to see. And then we have interferons, which again are chemical weapons and communication systems, as we're going to see. So the complement system, lots of things it can do, but the most important thing, the direct effect, is, no kidding, put a hole in the target cell. They even gave it a cool name. Membrane attack complex. Doesn't that sound pretty intense? Or MAC. So again, just Complement proteins doing the same thing as the purpurines did with the natural killer cell. Only these complement proteins are just circulating in your bloodstream as we speak. So again, put a hole and then let osmosis do its trick. The complement system can also act as chemotaxins or opsonins, those markers for death, and can initiate an inflammatory response. It can also help with the acquired immune system. Don't worry about this slide. It looks scary. I'm not going to ask you anything directly on this thing. But antibodies can bind to our target cell, and those antibodies provide sort of like a staging site for the complement system to bind and start their drilling process with their MAC. Don't worry about that. Interferons are kind of unique. We thought we found the magic bullet for cancer. It didn't quite works to some degree, but we're going to keep this really simple. What I want you to get out of an interferon, so there's several different types, but how does a virus work? Somebody impress me with how a virus works. Here's my cell. Sure. How does a virus infect a cell? What does it do? Okay. Yeah, not quite penetrate, but binds to the cell. All right. And so let's just make a nice little phage here. So it binds to the cell, and then it introduces in what? The nucleic acid. It could be DNA or RNA. And then it gets the cell to make the components to create more of the virus. Everybody with me? And please keep in mind that once a cell is infected, it's a goner. It never gets better. You can't fix a virally infected cell. Everybody got that? So this cell is a goner, but what it can do is send out interferons to another cell to say, hey, there's viruses out there, protect yourself. And then that cell that's not infected can produce things that stop the transcription process. And if you remember how this works to make a protein, you take the DNA and you make RNA, and from the RNA you can make a protein. Transcription, translation, and everybody knows that, right? Beautiful. If this cell is worn through an interferon, it can prevent the transcription translation process. Okay. For the more evil ones, retroviruses, why are they evil? Give me an example of a retrovirus. HIV. Another one that's evil? Herpes. Very good, herpes. And again, I'm not going to make this a genetics course, but a retrovirus does the exact opposite. It takes the RNA and makes DNA through what's called reverse transcriptase, and then they insert that DNA into your DNA, and so it's there forever. It's there forever. Okay. Can't get rid of it once it's there. Well, we're getting there. We're getting there slowly. A lot of your HIV drugs block the reverse transcriptase enzyme so that it can't convert the RNA into DNA. Everybody got that? So it was a little bit more of a micro and genetics lesson, but what I want you to be able to say is that interferons get the non-infected cell to produce enzymes to block the transcription process. Okay. Everybody got that? All right. This is a cool slide. Why do I like this slide? It's not just a picture, it's the whole picture, or the big picture. I know I've said that numerous times. Shows you how everything works here, so I like it. It tells a story. So first of all, we break through our physical defenses, and now we're inside. And before we can have a full-on infection, the queen bacteria has to enter the mother bacteria, the large one, that produces all the other ones. Is that correct? You guys learned that in micro? Lauren, learned that in micro? You haven't taken it yet? How convenient. You guys know I'm right? There's no such thing as a queen bacteria. You don't want to challenge me. I can tell you guys anything. And when you take micro, both you said there's a queen. Why is this a big bacterium? So they could put the word bacteria in it? What's that?
So I was thinking that, but I didn't want to say it.
That's okay. All right. Okay. Who's taking micro? All right, Candace. With all your micro skills and knowledge, what can you? Huh? Oh, well, you must have remembered something. I'm going to ask you very simple. How would you describe this bacteria from what you see? We got rod. Okay, that's good. Bacillus. What else could you infer?
It is not in a cluster.
No.
Attached. I don't know. I'm not sure where I'm trying to go.
There's one little other thing you could describe it as. How do you describe your basic bacteria?
By the color.
Yeah.
I mean, I thought that was just the illustration, but so it's the gram negative.
Yeah, I mean, that's what I'm saying. Yeah, that's all. The artist made it red, so it must be gram negative. Okay, gram negative rod. All right. All right. I'll leave the micro alone. Anyway, what I want you to get out of this is here we have an infection. Boom, we got the complement system is the crux here. It creates those membrane attack complexes to put holes in it. The complement system also acts as opsonins, marking the cells for death so our phagocytes can ingest them. The complement system also will activate mast cells that will produce the histamine and the chemotaxins, which will get our other white blood cells through that process of diapedesis to come out and do their thing. So everybody's working together. It's really, really cool. The only thing that we haven't covered yet is this little area right here, and that's the acquired immune system, and that's what we're going to talk about next. And then we'll have the full picture. Okay, so again, your acquired immunity means just that. You acquire it through exposures. You acquire it through exposures. It involves what is called cell-mediated immunity, i.e. cell-to-cell combat via the T cells, and the antibody-mediated immunity via the B cells. Again, we have to have antigens that will trigger response. It will activate T cells and B cells, and they both have their roles to play in immune defense. So, uniqueness of the acquired, it's very specific. I emphasized that last week, is if you're exposed to a particular pathogen, a virus, say, it gets you sick, you build an army, kill it, you get better. If you get a different virus, you're going to have to go through the whole process again. So these armies that you build after exposure are very specific to that particular virus or pathogen. It's very versatile. You can create antibodies for almost every type of antigen out there. And the key thing here is memory. You build a giant army of lymphocytes. You have different types of lymphocytes. Many of them are doing combat, if you will, but there is a select group that are memory cells. And they circulate in your body for almost ever. And if you're infected by that particular pathogen, it will stimulate those particular memory cells to create armies really, really quickly. So you can get infected by a virus you had before, but it's not going to get you sick because you're going to generate an army of lymphocytes that'll wipe it out before you even get a sniffle. Everybody got that? So by the time you're a full-grown adult, you probably have, depending on a lot of things, but anywhere from 20 to 60 different armies of memory cells for however many times you got sick in your life. Does that make sense? And again, we used to say they stay there forever, but we learned that, like many things I say in physiology, use it or lose it. So, for instance, it's the classic case of chickenpox. We wiped out chickenpox through vaccinations, but since there's no more exposure, ultimately those antibodies, or those cells, just died. So, as weird as it may seem, it's actually a good thing to be regularly exposed to chickenpox if you've already been vaccinated or if you're already immune, because that would trigger it. Doesn't really happen, but all of you that are going to go into the medical professions will probably have to get a series of. What's that? You're going to get vaccinations, but most of them are going to be, who knows the word? Boosters. Boosters. What does the word booster mean? You're going to boost those memory cells for that particular infection. So most of you have been vaccinated, but most of the things that are out there, you'll get booster shots because most of you haven't been exposed to that ever. Does that make sense? And of course, the key thing here is tolerance. We have to make sure that these lymphocytes don't start attacking your own cells, your own tissues. So your lymphocytes, like I said, started out as that multipotent hematopoietic stem cell that went on a different branch compared to red blood cells. But they all originate in the marrow. The B cells will stay in the marrow till they're fully mature. The T cells migrate to the thymus, where they will mature, and then they'll exit. Kind of just slightly mentioned that before, but once you become an adult, especially past age 40, 45, there's no real significance for the thymus to still be around. You have tons and tons of armies, so it kind of just atrophies a bit. Again, key role difference here, I like these are nice and simple. Your B cells are your antibody producing cells. Your grenade launchers. So they'll make antibodies that will bind to our target cell and hopefully neutralize them or destroy them. Your T cells are cell-mediated hand-to-hand combat cells. They will literally attach to the target cell and destroy it. Because they will work together. Introduced this earlier so I could use the word antigen, but again, remember antigen is anything that your body recognizes as foreign or your immune system will respond to. So an antigen will have binding sites, or epitopes, you might hear that in microbiology. Epitopes are antigenic determinants. Those are going to be particular proteins that are targeted by antibodies or receptors. So we did the blood lab and we talked about having type AB positive that would have three different epitopes, the A, the B, and the positive. Each cell is going, again, and I put them there because if you look at this artist, it makes it look like, hey, you just have a B cell and it's got, what, 6 receptors? No, there's thousands and thousands of receptors on one cell. Remember still, I emphasize this enough, it's not like those antibodies make a beeline to the bad guy. They're just floating around and they literally have to bump into the cell. So the more receptors you have out there, the more chance we have of binding. Again, slight differences. Here we have a B cell, and these are literally, they just put antibodies on the membrane surface, where the T cell has a unique receptor with one antigen binding site. We talked about it being versatile. You can create different antibodies or receptors for different antigens. What is changed or altered is this binding site. Okay? The rest of this stays constant. All right? So this is a protein, so it's got a unique chain of amino acids, but this can be rearranged to bind to our antigen. Thousands and thousands of combinations if necessary. So let's start with our C's, our T cells. Again, cell-mediated means the T cell is going to actually directly contact and try to destroy the target cell. They use all kinds of chemical signals. Cytokine is a very generic word for chemical signal. Cyto means cell. Kine, or kinetic, or kinesiology, means what? Movement, okay? Cell movement. We have cytokines called interleukins. Again, leukin, think of leukocyte, think of white blood cell. Interleukins is communication between white blood cells. So we already talked about the interferons. And then chemotaxins to attract us to the area. Now there's dozens and dozens of types of interleukins, so I decided to heck with it. I actually had, years ago, I had about the top 10 interleukins, but that was over the top. So you are just going to tell me collectively that interleukins do this down here. Initiate, mediate the inflammatory response, activate other B cells and T cells, stimulates the production of antibodies by the B lymphocytes, and stimulates more cytotoxic Ts and natural killer cells. Immunology is a fascinating subject. Very, very, very complex. It's a very, very simplified version of it. We have our 4 basic types of T cells. And under each one of those, if you were to take an immunology course, you find that there's several dozen types under each one of these, with different jobs and whatnot. This is the basic group here. So we have the cytotoxic T cell. That's the one that directly kills the target cell. Helper T cells activate Ts and B cells. So this is very similar to how you discuss the military. So you have the infantry, the direct, and the helper Ts are the guys at headquarters doing all the communicating. Then you have your suppressor T cells. Those are your military police, making sure no one's being a bad troop. And then the memory T cells, they didn't really fight, but they talk about the war. They were there. If you look under a microscope, you couldn't tell, you should have been able to identify a lymphocyte under the microscope compared to a monocyte, but there's no way you could tell if it was a B cell, a T cell, or any of these. The way we identified them was to find out what stuck to them, what markers stuck to them, and then we gave them classifications. So we'll keep this simple. So there are markers called CD3 markers that identify lymphocytes as T cells. All your T cells will have the CD3 marker. An additional marker called the CD8 is found on specifically the cytotoxic T cells and the suppressor T cells. Because they have jobs of destroying. And the CD4 marker is found specifically on the helper T cells. And I just introduced this because you're going to see this term in reference to what? CD4. Anybody? No one's ever heard the term CD4? In any way, passing? Well, that's disappointing. Now, if you're talking about HIV, they'll reference CD4 and helper T cells. Okay, here's the hardest part of this whole lecture. How do these cells recognize bad guys? What we have first is what we call markers that identify you as you, unique to you. We talked a little bit about this before. And that's your histocompatibility complex. Histo means what? Tissue. So yes. We take your histocompatibility complex and bind it with the antigen that we're concerned about. Collectively, we call this the MHC, or major histocompatibility complexes. There are two types of MHCs, class 1 and class 2. And you shall know them. Okay? So, class 1, pay attention. What you're going to have to provide me on a test is what cells are involved and what's the final result after interaction. So for class 1, MHC, we are talking about an infected cell, viral infected cell, or a cancer cell. What the infected cell will do is take a portion of the virus and stick it up on the surface, allowing T cells to identify the antigen. So again, this cell is, remember, once the cell is infected, as I said before, it's gone. It doesn't come back alive, doesn't get cured. All right. So think about that, because you really don't. But every time you get a sore throat and it's inflamed, a lot of those epithelial cells are infected. None of them are going to recover. They all have to die and slough off. The healthy ones eventually. Okay. So we have an infected cell. We put the antigen, the virus, up here with the class 1 markers. And now the cytotoxic T cell will bind to it. TCR just needs T cell. There's the CD8 marker. All right. So they combine with the class 1 MHC, and that activates the T cell. Again, the whole purpose of this is checks and balances, making sure this T cell isn't destroying a good cell. So it needs to see that antigen present. Once they bind, and this T cell recognizes this as an infected cell, it's going to destroy it. I said before, the best way to kill a cell is to put a hole in it. This is cool. This is a real pick. And again, the same process works for cancer cells. So here's a cancer cell, and there's a cytotoxic T cell. It's going to bind and destroy. Now listen to me carefully. This is another version, a little bit more complicated. But what I want you to get out of this is Here's my T cell. Here's my target infected cell. I am going to destroy it once I make the binding here. I destroy it by putting holes in it, perforins. I can release enzymes that will cause the cell to self-destruct. So one of those weird words, apoptosis. Everybody say it. Apoptosis, that means self-destruction. And other enzymes to just disrupt the metabolism. I want to kill this cell. Why do I want to kill this cell?
So it can spread.
Yeah, remember, I erased that too soon. Once the cell is infected, all right, with DNA and RNA, this cell is nothing more than a virus factory. All right, it's going to create viruses and viruses until this thing just erupts, dies, and then all those other viruses are released. Okay, so the key is destroying that cell before it starts producing a bunch of viruses. Everybody got that? Now, what's way important, put some stars, as many as it takes me to remember, that killing this thing is a small part of what ultimately has to happen. What ultimately has to happen is I need to start producing that army. So as soon as this thing makes contact, does its destruction, it's going to start cloning itself. Start dividing and dividing and dividing. And every single one of these newly created T cells are going to be specifically targeting that infection, those infected cells. So clonal means they're going to be identical. And this army of T cells I created are only going to work with that particular antigen. They won't have any effect on a different type of infection. We'll also make the memory T cells. So these guys will just hang out in case you get infected by this particular antigen before. Again. Class 2 MHC involves two different cells. We call the one cell an antigen presenting cell, or APC, antigen presenting cell. It can be a phagocyte, a dendritic cell, a macrophage. It's going to ingest our target pathogen. In this case, looks like we got a bacterium there. It's going to break it down, lyse it, and again stick a portion of the antigen on the surface. It's class 2 MHC. The cell that binds to it is a helper T cell. It has a CD4 marker. When they bind, the T cell is activated. The T cell, the helper T cell, does not kill anything. Its sole job is to alert other B cells and T cells that there's indeed an infection going on. It releases those interleukins. And we get this T cells stimulated for the attack and the B cells for antibody production. Again, different picture, same thing. So again, I have a macrophage, antigen presenting. It engulfs the bacterium, slices and dices it, puts part of the antigen on the surface. To clarify, when I say part of the antigen, it's a protein. It was part of the protein that made up this bacterium. All right, so it's basically a polypeptide. The helper T cell binds with its CD4 marker and receptor site, TCR, and that's going to release interleukins, once again, to stimulate T cell and B cell activation. So again, Class 1 involves an infected cell and a cytotoxic T cell. Class 2 involves an antigen-presenting cell and a helper T cell. And again, the key is clone, 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 clone army. I need lots and lots of these guys, and I need them to all be very specific for that particular infection.
Is the helper T cell the antigen presenting cell?
No. It's the macrophage. It engulfed this bacterium, broke it up, and now this macrophage is going to put a piece of the antigen on its surface. The helper is going to recognize it. It's going to recognize it. Everybody got that? Now don't move up the slides down. Again, the memory T cells, almost lifelong if you're exposed occasionally. If not, then yes, those will the booster ones. Don't worry about the thymosin, I'm not going to ask you anything about that. So, if you look at the big picture, where's the central focus? On the helper T cell. The helper T cell. And that's what makes HIV so bad, is most viruses are very specific for the type of cell, species, or organism. HIV directly attacks the helper T cell. It's exactly what you'd want to do in war. What's the first thing we're going to do in war if we're going to go fight an army? There you go. We're going to knock out their communications, their headquarters. You guys didn't know that? You guys don't watch any army movies? Yeah. And so that's exactly right. HIV knocks out the helper T cell, and now none of this works. None of this works. And so sometimes, what does AIDS stand for? Not autoimmune. Acquired immune deficiency. So if you think about what that really means, is it's saying your acquired immune system is deficient versus your acquired deficient. So you see what I'm saying? It's a little play there. But acquired immune deficiency means your acquired immune system is now deficient because I knocked out the helper T cell. HIV doesn't kill anybody. What kills an HIV patient? Is that? I'm sorry? They get sick.
Well, like they could get like a little.
They can't fight off.
Yes, they usually succumb to other opportunistic pathogens.